Bismillahirrahmanirrahim. Student, our today's topic of virology is the replication of viruses. You have already studied there are two types of viruses: RNA viruses and DNA viruses. We will cover this topic in two lectures. I am going to discuss some important things in our today's lecture. So let's start our today's lecture. Here you can see objectives of our today lecture. You are going to understand viral growth curves, cytopathic effects of on affected cells of host replication, some key points of replication, and some initial steps like attachment, penetration, and uncoating of viruses. And we will also discuss a few things about gene expression and genome replication of the viruses. Usually, it is observed that a common virus is able to multiply into hundred of virons in average 10 or in animals, especially in animals. So human viruses or animal viruses can take many hours to replicate into the hundreds of virons, and on the other hand, some important plant viruses or bacteria viruses undergo replication within few minutes. Here another important thing is eclipse period. What is eclipse period? And the initial at the initial stage of virus replication, virus undergoes some the time during no virus is found inside this cell. During after attachment to the cell, when cell is undergoing toward replication, it means when nucleic acid of the virus is attached with the nucleic acid of host cell, and the cell is in the dividing stage, then you can observe there will be no cell in the host cell, no virus in the host cell. And you can say then uh, you can say there is no virus in the cell. But after few hours, when replication or when the protein formation started completing or starting near to the completing, then when virus count getting raised into the into in this host cell. During early stages, when viral nucleic acid attaches with the nucleic acid of host cell, then a nucleic acid of host cell, then virus become disappear in the cell. But nucleic acid of virus is attached with the host genome, and the viral genome is getting going to double or triple soon. But in initial stages, when a complete virus is ruptured, then virus is then initial period when we cannot observe any virus in the cell due to rupturing of cell, due to rupturing of virus, because virus needs only nucleic acid for doubling. A complete virus will be disappear. So this phase is known as eclipse period. And after a few hours, when virus getting when formation of virus is going to complete, then virus count will starting raising in the cell. So we is then around after 10 hours there will be we can observe hundred of viruses from one virus inside the cell, or after rupturing or release of viruses from the cell. So the eclipse period is actually a period when we cannot an initial stages where we are unable to observe any virus in the nucleic acid, because any virus in the human host cell, because virus was denatured because for the purpose of replication. So we can discuss in this stage in our final summary. Now another important thing is latent period. The eclipse period end with the appearance of virus. The latent period, the eclipse period actually ends with the within 1 to 2 hours on the other sorry the eclipse period end in the 1 to 4 hours, but on the other hand there is another period is latent period. This is actually a time from the onset of infection to the appearance of virus extracellularly. It means after release of hundreds of virus, a time from zero, or a time from infection, entry of viron into this host cell, to the release of virus, the whole time. It means 10 hours is the latent period for a virus. If virus is going to release within the 10 hours, then this period is the latent period for the virus. So another important thing is virus. There are hundreds of virus replicate from a single virus. Another important thing is when the replication was occurring in the cell, we can observe some important effects on the host cell. When we can observe some important effects on the host cell when virus gets entry into the host cell, and viral genome gets attached with the host genome, then a host cell undergo the influence of some important non-structural protein of the virus. These non-structural proteins are actually some enzymes of viruses, or actually some other supportive structure which is present inside the cell or inside the core of the cell. And the important first important cytopathic effect is the change of ATP influence. It means when replication of viruses get start, then ATP which was using by a host cell for metabolism or for energy purposes, ATP gets influenced by virus cells. So virus use our host cell ATP for replication, for own replication. So ATP production gets deficient in our affected cells. And the another important thing is change in genome. When virus attacks the host cell, then the viral nucleic acid attaches with the genome of the host cell. This is known as change in the genome of the host cell. So when genome of the viral when genome of the host cell gets interrupted or contaminated with the virus of the genome of the RNA or genome of the virus, then we can say this is a change in the genome of host cell. So this is also a cytopathic effect. And other important thing is change in working of enzyme. So we have host cell have many enzymes for metabolism for many chemical reactions, but due to attack of the viral proteins or virus nucleic acid, working of enzyme become distorted. So enzymes working of enzyme is working of enzyme is very important for normal metabolism. When ATP production has been damaged for from viruses, then working of enzymes is also interrupted by viruses. Then another important factor is cytopathic effect. Our few cells are responsible for the production of natural immunoglobulins, which are known as interferons. Interferons attacks on viruses to kill viruses. We have naturally an amazing or golden molecule which is interferon. Interferon attack on all type of viruses to kill them, but some viruses can cause the switching of the gene which is responsible for the production of interferon. So due to the switching of the gene of interferon production gets damaged, and due to deficiency or non-production of interferon, viron production gets increased with the time. Another important cytopathic effect of viruses is the hindrance or in the stoppage of the metabolism of the cell due to damage of ATP production, due to damaging of enzyme, many enzymes, metabolism of the host cell also damaged. So these are all are important cytopathic effects on the host cell by the virus. Another and the last ultimate, due to as you know, change in nucleic acid or change in enzyme or change in energy source of the cell can lead to the death of the cell. So ultimate fate of the viral infected cell is death. So it is also a cytopathic effect of the virus on the host or infected or attacked cell. So here we are going to start. Our major topic is replication of virus. We have already studied in the case of bacteria. Bacteria divides on the as the on following the role of binary fission and other few methods. And mitosis occurs in usually human cells. Mitosis meiosis occurs in usually eukaryotic cells. And another important thing is the replication in the virus. Virus replication is different from all other eukaryotic or prokaryotic cells, because virus is also different from all other cells in many ways. So first few steps are attachment of virus to the host cell, and the attachment and then penetration of virus into the host cell. And after penetration, virus proteins virus coat or capsid and viral nucleus get separate from each other due to the action of cell vesicle, host cell vesicle. And the RNA or DNA, it means genome of the virus become active, and the copies of genome genes means RNAs or DNAs started production. So gene expression. Another important step is gene expression. Some important genes of viruses shows expression, and important proteins forms. Then replication complete. After completion of replication and the production of structural proteins or enough copies of viral DNA. sorry, viral genome, viral genome and capsid forms within the cell at the final stage of the, it means at the final stage of the viral replication we can observe many maximum copies of viruses in a single cell. So after assembly of viruses or after complete achievement of the shape by virus gets released into blood or fluid or extra or any other species by bursting the or by rupturing the cell. So many so commonly when virus attack the cell when virus attack the cells, the ultimate fate of that cell is death. So then when hundred of viruses released from the one infected cell, so these hundred of viruses go for the infection of other similar cells, लाइक कि अगर एच आई वी के अंदर एच आई वी का जो वायरस है वो क्या करता है, अटैक करता है किसी टी लिम्फोसाइट पर, उसको रप्चर करके अपनी 1 से 100 कॉपीज़ बना लेता है, तो वो 100 कॉपीज़ अगली दफ़ा में जाकर 100 टी सेल्स को डैमेज करेंगी और उन 100 से फर्दर 100 कॉपीज़ निकलेंगी. सो इस तरह वायरल लोड जो है वो बढ़ जाता है. सो काउंट ऑफ वायरस इन आवर बॉडी इज़ नोन एज वायरल लोड, क्लिनिकली इज़ नोन एज वायरल लोड in a patient. So in case of coronavirus patient we have to control actually viral load. So antibodies of our bodies, after few days antibody production of from our body get started against any type of viruses, and they can the antibodies of our body can kill virus efficiently, but the major problem is the damaging of lungs tissue due to coronavirus. Actually damaging of lung tissue is not due to coronavirus, it's due to our own body response, our own body mediators can kill our lung tissues or lung cells. So when inflammation when after attacking the coronavirus attacking the coronavirus, inflammatory mediators released when these inflammatory mediators attacks the lungs tissue as well. So these inflammatory mediators are actually so strong that they can unintentionally kill the lung tissue. Killing of any tissue is harmful for our body. We can save our tissue with anti-inflammatory drugs as well. So another we are going toward another important slide, which is the actually replication overview of replication steps. So here is a question is what are the important replication steps, a common important replication steps. So there are major replication steps are attachment and the penetration by the parental viron. One single viron which is going to infect another cell is parental viron. So when parental viron attached with the host cell, then by the action of cell or by the action of some enzymes of virus penetrate into the host cell. When virus penetrates into the host cell, then low pH of the vesicles, especially when वेजिकल देखें, जब कोई भी चीज़ सेल में एंटर होती है तो उसके अराउंड एक वेजिकल बन जाती है, ठीक है. सो आई विल डिस्कस दीज फिनोमिनाज इन लेटर. सो आफ्टर अनकोटिंग ऑफ वायरल जीनोम, viral mRNA viral mRNA synthesis occurs when mRNA forms many proteins. Viral proteins, which are actually initial proteins, actually are enzymes. So many enzymes are formed from the mRNA of the virus. So these viral proteins and these viral RNA attach with the genome of the host cell, and the viral proteins, and with the viral proteins, viral genome gets replication by attaching the uh, genome of viral RNA with host genetic material. In simple wording, actually replication is difficult one. There are I am going to simplify it in simple wording. mRNA of virus, the after uncoating जब न्यूक्लिक एसिड निकलता है तो न्यूक्लिक एसिड ऑफ वायरस जो है वो होस्ट में आकर दो काम करता है. पहला काम ये करता है कि या तो वो mRNA की सिंथेसिस करवाता है. mRNA की सिंथेसिस से प्रोटीन बनती हैं. ये प्रोटीन एक्चुअली इंजाइम्स होती हैं. वो इंजाइम जिन्होंने वायरस के आरएनए या डीएनए की रेप्लिकेशन करवानी होती है, ये इंजाइम्स जो बने हुए होते हैं, और वो आरएनए जो होस्ट के डीएनए के साथ अटैच होता है, होस्ट के डीएनए या आरएनए के साथ अटैच होता है, वो इन इंजाइम्स की मदद से उसकी कॉपीज तैयार करता है. सो आफ्टर द कंप्लीशन ऑफ एनफ जीनोम फॉर्मेशन ऑफ वायरस, लास्ट स्टेप पर एक और वायरस जो आरएनए बने होते हैं वो दोबारा से क्या बनाते हैं, वो और प्रोटीन्स बनाते हैं. दीज द लेट वाइटल प्रोटीन्स आर एक्चुअली स्ट्रक्चर प्रोटीन ऑफ द वायरस. दीज स्ट्रक्चर प्रोटीन्स आर द पार्ट्स ऑफ कैप्सिड ऑफ द वायरस. सो जो थर्ड लास्ट स्टेज पर आपको प्रोटीन नज़र आ रही है वो एक्चुअली स्ट्रक्चरल प्रोटीन बनती हैं. सो पहले में इंजाइमेटिक प्रोटीन बनती हैं, देन लास्ट पर स्ट्रक्चरल प्रोटीन बनती हैं, और यहाँ तक पहुँचते हुए वायरल जीनोम जो है उसकी एनफ कॉपीज़ जो हैं वो रेडी हो चुकी होती हैं, तैयार हो चुकी हैं. सो जन क्या होता है, प्रोजनी वायरोन असम्बली. इट मीन्स वायरस की जो शेप है उसके अकॉर्डिंग वायरस की प्रोटीन और आरएनए जो है उसको जोड़ा जाता है. सो एट द सेकंड लास्ट स्टेप ऑफ means at the assembly step of the virus, you can observe maximum number of viruses in a single host cell. And after completion of the shape of the viral cell gets ruptured, and number and many viruses from a single viron released into the blood or plasma, etc. So we can discuss all these steps in later. First few steps are attachment, penetration, and the uncoating. You should know the structure of the virus very well. First of all, first step is the attachment of virus to the host cell. Actually, what is the attachment? We know, we have already studied that a virus contain spikes on the surface on capsid, or many other viruses contain many attacking or adherent proteins on their structure. So when virus attached on the receptors on specific surface, you should know that virus is specific to receptors and specific to cell as well, and some viruses are also host specific. With the point of host specific, we have divided viruses into two steps: narrow range viruses and the broad range viruses. Narrow range viruses actually those viruses which can which can cause infection in a few hosts, like poliovirus can infect only humans or some primates. On the other hand, there are a broad range of viruses like rabies viruses can cause infection in many species or many mammals, like bats, in dogs, in humans, etc. So this is actually two terminology. These are actually two terminologies important for you. So actually, when virus gets attached to the host cell, then they form weak covalent bond with the host cell. So After attachment structure after attachment with the structure protein through the structure protein with the host surface of the host cell surface, then non-covalent weak non-covalent bond forms, which this weak covalent bond helps the virus to enter into the in and to enter into the cell, there is another phenomena occur which is known as pinocytosis. After attach, there are few examples of attachment specificity according to specificity to the receptors, like herpes simplex virus type 1 attached only with the fibroblast growth factors receptors only, and the rabies virus attached the acetylcholine receptors, and the HIV virus attack on CD4 receptors on the surface of T lymphocytes. Epstein-Barr virus binds with the complementary receptors, and the vaccinia virus binds on the receptors of epidermal growth factors. So here are some examples according to specificity of the virus toward the receptors. So viruses are specific to receptors and host as well. So you should must keep in mind that there are some important examples are rabies, HIV, and herpes simplex. So we are going to deal step by step all few important point. First of all is attachment. We have done with attachment of the virus. After attachment, we you can observe in diagram number 1 virus. After the attachment of virus, there is a groove which forms on the cell of on the host cell, and the after the formation of groove, there is a phenomena similar with the phagocytosis. You have studied in first year that the how phagocytosis occurs. Membrane of the host cell form engulf the actually engulf the virus. So virus comes inside the host cell in vesicle. So you can observe in the diagram number 2, virus in the present in the vesicle inside the host cell, and after a few time or suitable time, or after attachment with the suitable after when a virus when vesicle achieved low pH, then due to low pH or acidic pH, viral structure gets distorted, or virus gets disrupted. So due to destruction of the viral structure, viral protein and genetic material gets released into the viral contents are released into the inside the cell, and viral structure has been distorted, and you can observe the genetic material inside freely moving genetic material inside the host cell, and the other structural protein. Now but these structural proteins are not enough to cover a new increased number of replicated viruses. So there is a need for the formation of more structural protein. So this genome of the this free genome of the virus undergo the formation of mRNA, because protein synthesis occurs through the from messenger RNA. So importance of messenger RNA formation is much important. So there are two important points. First one is the pinocytosis is actually is actually the penetration of the virus, and the another is uncoating due to low pH. So after uncoating, nucleic acid gets released into the cytoplasm of the cell, and it depends upon the cell के वायरस का जो जेनेटिक मटेरियल है, डीएनए या आरएनए, वो वायरस के न्यूक्लियस में डिवाइड करेगा या वायरस के सेल में डिवाइड करेगा। मेरी बात की अगर नहीं समझ आई तो हम नेक्स्ट स्लाइड पर चलते हैं। वी विल डिस्कस इट इन दिस व्हेन वी कवर अदर स्टेप्स ऑफ दिस। Here is another important thing is about attachment and penetration coating. In bacteriophages, those viruses which can cause infection in the bacteria. In case of bacteria, viral genome. Just viral genome enters into the cell. Structural protein remains outside of the cell. You can observe in the diagram, in the diagram in at the right side of the diagram you can then, when bacteriophage attack on virus, when bacteriophage virus attack on bacterial cell wall, and with the help of bacterial receptors and with the help of tail from virus, both become in contact, both came in contact, and after binding or after suitable bind or after suitable binding, virus viral there are lysosomes enzyme released from the virus. When these lysosome enzymes causes destruction of the viral bacterial cell wall. These bacterial cell wall, when bacterial cell wall gets disrupted, then DNA of the virus was pushed from the uh, virus so into the bacterial cell. So when the viral DNA pushed from the viral protein into the cell, so this DNA, this DNA can cause infection in the bacteria. So there was another discussion about the infectivity of the virus nucleic acid, डेट के जो वायरस का न्यूक्लिक एसिड है वो इन्फेक्शियस है कि नहीं है। सो हेयर यू कैन ऑब्जर्व डेट स्ट्रक्चर प्रोटीन्स वाज लेफ्ट बिहाइंड, लेफ्ट at outside, but only viral DNA or nucleic acid was entered into the cells. So after penetration of viral DNA into the bacterial cell, it can bind with the nucleic acid of the bacterial cell, and after proper or suitable time, number of copies formed from the single viral DNA. So this is the actually we can say that nucleic acid is also infection. Viral nucleic acid is also infectious agent. So there is an important step in the replication of the, or in the gene expression of the genome replication. mRNA, first step is the viral gene expression is mRNA synthesis. It means mRNA synthesis is important for the manufacturing of structural and non-structural protein of viruses. You have already studied that virus has only two structure, nucleic acid and protein, nucleic acid and coat of protein. So without protein coat virus, virulence of virus is not enough for suitable infection or for proper infection. So first step is the formation of mRNA. mRNA synthesis is necessary for the production of structural and non-structural protein. So the first step is the formation of mRNA. mRNA synthesis is can vary with the nature of the genetic material. In case there are two types of RNA viruses. Um, there are positive strand RNA viruses, and there are negative strand RNA viruses. So in case of positive strand RNA viruses, replication or replication of mRNA starts from the from initial step. It means positive strand RNA can directly bind with the host ribosome, but in case of negative sense RNA, negative sense RNA virus, uh, viral viral RNA viral RNA is unable to attach with the ribosome of the host cell. Without binding with the ribosome of the host cell, viral RNA cannot undergo protein production. So, attachment with the ribosome of the host cell is necessary. Positive sense RNA strand viruses can directly bind with the ribosome of the host cell, but negative strand RNA viruses are unable to attack on ribosome. So we will discuss later why this is occurring in the RNA synthesis. So here is another important thing is gene expression, about gene expression and gene replication in according to DNA viruses. We will discuss the nature of gene expression in DNA and RNA virus separately. In DNA viruses there is a simple mechanism, because mostly host cells have genetic material in the form of DNA. So after penetration or after uncoating of the virus, when viral DNA released into the cytoplasm of the host cell, this DNA of the virus gets entered into the nucleus of the host cell. So most of the DNA replication, DNA of the viral DNA replication of the viral DNA occurs in the nucleus of the host cell. Please keep in mind that DNA replication, mostly DNA replication occurs in the nucleus of the host cell, and the RNA replication of RNA viruses occurs in the cytoplasm of the host cell. But there are here a few exceptions also here. Exception is pox viruses. Pox viruses are DNA viruses, but due to lack of some enzyme, or they have their own polymerase, so they divide, their genome divides in the cytoplasm of the cell. And on the other hand, there are some double stranded DNA. Mostly all DNA viruses have double stranded DNA in genome or in the form of nucleic acid, but parvovirus is a virus with single stranded DNA. So we you should keep in mind that DNA replication occurs mostly in the nucleus of the host cell. Uh, Now gene expression. Now how gene expression can occur in RNA viruses. RNA, I have told you in previous slide that replication of RNA viruses occurs mostly in cytoplasm of the host cell. So here are some also few exceptions. Retroviruses family and influenza viruses. Retroviruses, they are are few important steps in the replication of retroviruses and influenza viruses. So they entered into the nucleus. They entered into the nucleus of the host cell to complete their important steps, but most of the RNA viruses completes their replication in the cytoplasm. All RNA viruses are single standards. Standards virus, all RNA viruses are single standard in nature, but there are some few families like Rio virus family, also known as the retroviruses family. Retroviruses also part of the Rio viruses family. So all viruses, mostly viruses are RNA viruses are single stranded in structure. So actually on the basis of the replication, there are few changes in the replication, few steps of the replication in the RNA viruses. So we have divided RNA viruses in four groups on the basis of change in replication steps. I'm going to discuss these four groups in few words in next lecture. In our next lecture, we will discuss these groups in details. There are four groups on the basis of the replication of RNA. RNA of the viruses are mostly single stranded, and few single stranded RNA viruses have positive polarity. It means they are known as positive sense RNA viruses. So these what are the function of these positive viruses? They can directly bind the RNA of positive polarity ribosomes. Positive polarity viruses can directly bind with the ribosomes of the host cell. So after binding with the ribosomes, viral RNA can undergo replication and transcription, can undergo the formation of protein in simple wording. So protein formation can occur in single stranded RNA viruses quickly as compared to other viruses, because few steps are missing, and the viral RNA is directly undergoing the formation of viral protein. So when viral protein form, coronavirus is also single stranded positive with positive polarity RNA virus. That's why the incubation period of this coronavirus is very low, actually 14 days, 2 to 14 days. So you should also aware about our pandemic, this pandemic also. So RNA mRNA, when mRNA formation directly occurred with the use of RNA, initial RNA from virus, this is actually easy for the virus to cause early disease. Then another important group is single stranded RNA viruses with negative polarity. Those viruses which have RNA with negative polarity cannot cause directly protein synthesis from RNA. So they undergo the, first of all, they have to go for the mRNA must be transcribed by using RNA strand. So RNA of the genome of the virus, initial RNA of the virus transcribed into the new RNA. This new RNA is actually a messenger RNA. So, though after formation of messenger RNA with the help of RNA polymerase, by keeping initial RNA stand as a template, this viral new mRNA virus undergoes a protein formation. So protein formation is very necessary to cause the disease. So basically the difference is in the first few step of the mRNA formation. mRNA formation in positive sense RNA viruses occurs directly from first step when RNA genome directly binds with the ribosome and they forms messenger RNA, then messenger RNA forms proteins. But in case of negative sense or a negative single stranded RNA viruses, first of all formation of mRNA is necessary, but mRNA formation but RNA of the but RNA of the negative RNA of the virus is unable to detect ribosome of the host. So after when RNA when RNA is formed from the genome or RNA of the virus, initial RNA of the virus, this viral RNA gets attached with the ribosome of the host cell and can cause protein production or etc. Another important thing is double stranded RNA as genetic material. Double stranded RNA as genetic material, it means RNA has a double strand in which we can there mostly in double stranded RNA viruses, one strand is positive and the other strand is the negative strand but in case of rotaviruses, but these positive strand is not able to replicate directly into mRNA synthesis or is unable to form mRNA. And so but because positive strand and negative strand RNA are strongly attached with each other with the help of hydrogen bonds. So some important diseases can cause in children like diarrhea. Common rotaviral disease in children is diarrhea. Another important group is single-stranded RNA or positive polarity, means retroviruses. These viruses are actually are like our first group, which is single-stranded RNA with positive polarity. But in case of retroviruses, there another enzyme is present in the viruses, which is known as the reverse transcriptase virus reverse enzyme. Reverse transcriptase enzyme has the ability to transcript what is the transcription? First of all, you should know about the transcription is actually the formation of RNA from the DNA. So DNA stand used as the a template and new RNA stand formed. So from RNA formation from DNA is actually a transcription, but the formation of DNA from RNA is known as reverse transcription. So because retroviruses have RNA in as their genome, so when RNA and when RNA these this RNA is unable to directly undergo the formation of protein. So first of all, this viral RNA undergo the production of DNA. So DNA preparation occurs from the RNA by use of special enzyme from virus, which is known as reverse transcriptase enzyme. This reverse transcriptase enzymes convert RNA into DNA, and after the formation of many copies of DNA, all copies of DNA forms many copies of RNA. So then RNA forms many proteins or structural and non-structural proteins. So all proteins in the final step of the virus can cause assembly and the release of the virus from hundred of viruses from a single virus. So we will discuss these steps in easy wording in our later session. So this actually replication is difficult to understand because we have not studied some important things about viruses. So we will discuss these all these steps in our live session as well. So for a time period for our today's lecture it is enough, but replication is not complete yet. We will discuss all other and last steps of the viruses and some important steps of the viruses in our next lecture. So take care.